Hello everyone, in this video I'm going to speak about Agarose gel electrophoresis, which is not a new technique. It's a very old technique, but it's still used until nowadays because it's a very useful technique and very important one. So let's start to ask what is Agarose gel electrophoresis. Agarose gel electrophoresis is a technique used to separate nucleic acids, DNA or RNA samples. Um, it uses agarose matrix gel, which is composed of agarose, which is a kind of sugar, and when the agarose molecules are bound together, they will form something like a porous matrix or porous network, which is the agarose gel. It looks like this. So the pore size in the agarose gel depends on the concentration of the agarose solution. I will speak about it in a few minutes. So first of all, let's speak about how to perform agarose gel electrophoresis. To perform agarose gel electrophoresis, first we should know how to prepare the gel. To prepare the gel, we need agarose, we need the buffer, which is trace borate EDTA buffer, or what we call it TBE buffer of pH 8. We need a visualizing dye, which is normally what we use is ethidium bromide, but since ethidium bromide has been found to be very highly carcinogenic and a very dangerous material, some people decided to substitute ethidium bromide with other visualizing dyes like cyber green or others. What we do is that we take the agarose powder, which looks like this, and we mix it with the buffer, trace borate EDTA buffer, and we dissolve it. To dissolve the agarose in the buffer, we need to boil it. So we boil it in the microwave. Why do we choose the microwave? Because it's faster and because in the microwave the solution doesn't evaporate, so the concentration will not be changed. Once the agarose is dissolved in the buffer, we add the visualizing dye. So the gel at the end will look like this, as I told you before, and the pore size depends on, as I told you, depends on the concentration of the agarose solution. Now, what we do is that we take this solution and we pour it in a plastic container like this, and we add this plastic piece in order to create some wells in the gel where we apply our samples. The gel takes 20 to 30 minutes to solidify, and once the gel is solidified, we put this plastic container in a chamber, and we fill it with the same buffer, with trace borate EDTA buffer, the same buffer we used in the gel. And then we remove this plastic piece, so we end up with a gel looking like this. So this is the gel from above, this is our gel, these are the wells when we apply our DNA or RNA samples. What we do then is that we apply our DNA or RNA samples in these wells, and then we apply an electrical current using a battery or a power source, applying a negative charge toward the samples and a positive charge on the other side of the gel. What's going to happen is that nucleic acids are going to migrate from the negative toward the positive charge. Why? Because the nucleic acids are negatively charged. Why the nucleic acids are negatively charged? Because of this, because of the phosphate. So we know that nucleic acids, either DNA or RNA, are composed of sugar, either ribose or deoxyribose, and the nitrogen base and phosphate groups. So the phosphate are negatively charged, and because of this, any sample of nucleic acids, either DNA or RNA, is negatively charged. And because of this, the nucleic acid samples will migrate from the negative toward the positive charge. Now, what we should do is that first of all, we should prepare our sample. The sample of DNA or RNA is extracted from a cell. So either we extract the DNA or the RNA. Then we mix the sample with loading buffer and with the restriction enzymes. I'm going to speak about the two of them. First, the restriction enzymes are very specific enzymes. They are endonucleases, so they cut the DNA And this thing is that why they are called restriction, because they cut the DNA in a very, very specific place. So there are many restriction enzymes, but each one of them can detect a specific sequence of the DNA, specific sequence of nucleic of nucleotides. So let's say one restriction enzyme can detect the sequence of CCGT, for example, and then once this enzyme see the CCGT, it cuts downstream the sequence. So each restriction enzyme can detect a specific sequence and cut downstream, downstream this sequence. What we're going to have then is smaller pieces of DNA. Why do we apply restriction enzymes? Is because the DNA is a very long chain, so we cannot apply it in the gel, but we need to cut it first. The loading buffer is composed of glycerol and coloring dye. First, the glycerol is used to increase the viscosity and then the density of the sample. Why? Because once we apply the sample in the well, we want the sample to settle down. Otherwise, it will be mixed with the buffer and then we will lose our sample. But to increase the viscosity, when we increase the viscosity and then the density of the sample will settle down in the well and will not be mixed with the buffer. The coloring dye mostly used is the bromophenol blue. It's a blue dye. It's used to give the sample a color, so we can detect the sample on the gel once it migrates. We can see that the sample, for example, reaches the end of the gel, and then we turn off the power source. Now, how does it work? Let's say that this is our gel. The well will look like this from a side look. So what we do is that we apply our sample in the well, so it will look like this. And then we apply the electrical current. So the sample will start to migrate from the negative toward the positive charge. The thing is that the large sequences of DNA will stick to the gel earlier because it depends on the pore size, because they cannot migrate farther in the pores. But the smaller sequence will migrate farther, and the smallest sequences will migrate the farthest. Now you can imagine it this way. So let's say that this is a gel. There are pores inside. As I told you, the pore size depends on the concentration of the agarose buffer solution. So yeah, the larger sequences cannot migrate farther, but the smaller sequences can migrate farther. Now, why is agarose gel electrophoresis used, or what is it used for? So let's say we have different DNA samples from different individuals, we will get different patterns. Some of the sequences or some of the bands will look the same, and some of the bands will look differently. But why do we use it for? Of course, there are many applications of agarose gel electrophoresis, but now I want to speak about three of them: forensics, relationships, and medical diagnostics. First, let's speak about DNA fingerprinting. What is DNA fingerprinting? DNA fingerprinting is it's exactly like a fingerprint. So if we take a DNA sample from any individual and we apply it on agarose gel electrophoresis, we will get different bands from another individual. Why? Because there are something in the DNA called the DNA junk sequences. What are these sequences? They are junk. From their name, they are junk. They are non-coding sequences. So they are not genes or anything. They are junk sequences. They are short repeats. So they are non-coding sequences, short repeats, and the most importantly, they are unique. So each individual has junk sequences which are different from another sequence. So let's say we have individual 1, which had this junk sequence, GCC, GCC, GCC. So it's a short repeat. It's GCC repeated 5 times. Okay, and let's say we have individual 2. So individual 2 had the same repeat, but it's longer. Another individual might have another repeat, or it might have the same repeat, but it's longer or shorter. The thing is that it's different. It's different in each individual. So these individuals might have some identical sequences like this. For example, these sequences are identical between individual 1 and 2. They might also have some similar sequences like these, but they are not identical. And they will have the junk sequences which are totally different. So the thing is that if we take DNA samples from individual 1 and individual 2, and we apply the same restriction enzyme in both of them, the restriction enzymes will cut the DNA differently, and so we will get different bands on the agarose gel electrophoresis. Now the question is, how can we apply this in the forensics? Let's say we have a crime, we have a sample from the crime scene, and then we have two suspects, individual 1 and individual 2. We apply the DNA samples from individual 1 and individual 2 on the gel, so we get bands like this. So they are some identical bands, and they are some different bands, which are the junk sequences. This sample is taken from the crime scene, so it might be taken from a hair or anything found in the crime scene. Then when we apply this sample on agar gel electrophoresis, what we are going to see is that this sample is exactly identical to individual 1, and then we can say that individual 1 is the criminal. Well, what I want to tell you is that, of course, we need to treat the three samples with the same restriction enzyme, because if we apply different restriction enzyme, we will get different pattern. So when we want to see, like when we want to compare samples, DNA samples, we need to treat them with the same restriction enzyme. Well, we can use the second application I want to tell you is relationships. So let's say we have individual 1 and we want to see which one of individual 2 and 3 is the father of individual 1. What we do is that we apply the DNA sample from 3 of them. As I told you before, again, we treat the samples with the same restriction enzyme, and then we apply them. What we see is that we have some identical sequences in all of them, and we have some sequences in common, and we have some, like for example, individual 1 had this specific sequence which doesn't exist in, or band. Why I'm saying sequences is band. So we have some identical bands. Individual 1 has this specific band, but if we compare, we see that individual 1 has common bands with individual 3, like this one, for example, this one, and this one. So individual 1 has more common bands with individual 3 than with individual 2. Then we can say, yes, individual 3 is the father of individual 1. What we can also use the agarose gel electrophoresis for is medical diagnostics. So if we have an individual that we are doubting that he has a mutation in gene A, for example, how can we ensure about this? We get a normal allele. So this, we know that this allele is this sequence or in this gene is normal, and we treat the two of them with the same restriction enzyme, and then we apply them. Well, we see that we take this identical bands again, but then gene A will look differently in allele 1 and allele 2, and then I can say, yeah, there is a mutation here. Now let's see how can we visualize the gel. As I told you, for the visualization, we use this visualizing dyes, which we mix with the gel. I told you we have ethidium bromide, cybergreen, or others. There is also methylene blue. These visualizing dyes can bind effectively with the DNA or with the RNA. Cybergreen can bind only with DNA because cybergreen can bind with a double strand. It needs a double strand, so it binds only with the DNA. Ethidium bromide can bind with RNA and DNA. So so when we apply the DNA samples, these materials which are inside the gel, of course, one of them, we use one of them. These materials bind to the DNA bands, and then they are fluorescent, so they give like fluorescence. under certain wavelength. For ethidium bromide, it's UV. For cyber green, it's another wavelength. So here it is. This is ethidium bromide. It gives fluorescence under UV light. This cyber green, it emits light under 497 nanometers wavelength. So this is everything I wanted to tell you about agarose gel electrophoresis. I hope you enjoyed the video. If you really enjoyed it, don't forget to like, share with your friends, subscribe the channel, because I really need this. Then if you have any questions, you can write in the comments. I will answer you. If you have any suggestions for other videos or other topics I can speak about, you can also write in the comments. And see you in the next video. Bye.